Welcome to our next segment of the chemistry of beer. The topic for our discussion today is glycolysis. Glycolysis is a very interesting pathway, and it should be relevant for us to help us understand how yeast process glucose, but it also applies to us. It's an ancient pathway, and it is common to both yeast and humans. To begin our discussion of glycolysis, we'll first of all think about the big picture, and we'll answer the question, what does glycolysis do for an organism? Next, we'll consider sources of glucose, and in particular, we'll be thinking about yeast. Then we'll move on to a discussion of the overall pathway, and then finally, we'll tie up some loose ends and summarize what we've discussed. Before we dig into the details of glycolysis, it really helps to take a step back and think about the big picture. To begin, let's define what glycolysis means. It literally means sweet loosening or splitting. As I noted, it's a very ancient pathway and it's present in nearly all organisms. The net reaction, which you should know, is as follows. We start with 1 glucose, 2 NAD+, that's our old friend again, 2 ADPs, 2 adenosine diphosphates, and 2 PIs. PI stands for inorganic phosphate. At the end of the pathway, we form 2 molecules of pyruvate plus 2 molecules of NADH plus 2 molecules of ATP. As I've mentioned before, ATP is sometimes called the energy currency of the cell. So this pathway produces ATP, which the cell can use to power its many functions. What are the sources of glucose? As you learned in an earlier part of this course, some of the initial steps of the brewing process yield glucose and maltose. If we look at our diagram, we see that the glucose can enter the cell through a glucose transporter. And we see that the maltose can enter the cell along with the proton. That is called symport. So there is a maltose hydrogen ion or maltose proton symporter that brings in maltose and the hydrogen ion. The maltose can then undergo a reaction catalyzed by the enzyme maltase and form 2 glucose molecules. So now we are ready to discuss glycolysis. Shown in the diagram is the entire glycolytic pathway, as it is sometimes referred to. If we look at the upper left-hand corner of our diagram, we see that we start with glucose. Now the first reaction converts glucose to glucose 6-phosphate. This reaction is catalyzed by the enzyme hexokinase. Kinase is a general term for an enzyme that catalyzes what we call ATP-dependent phosphorylations. So you see above the reaction arrow, ATP is being converted to ADP plus a proton, and a phosphoryl group has been transferred from ATP to glucose to make glucose 6-phosphate. One of the things that this reaction does is it helps keep the glucose inside the cell, because in its phosphorylated form, it cannot pass through the glucose transporter. The next reaction is an isomerization reaction, and it converts the glucose 6-phosphate to fructose 6-phosphate. The third reaction is another kinase reaction. The name of the enzyme is phosphofructokinase, and it catalyzes the ATP-dependent phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. So now we have phosphoryl groups attached to the number 1 carbon and the number 6 carbon. What we've essentially done is made a more symmetrical molecule, and that will make sense when we consider the next reaction. The next reaction is the aldolase reaction. Aldolase catalyzes the cleavage of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These 2 molecules themselves are interconverted by another enzyme called triose phosphate isomerase. And it is the glyceraldehyde 3-phosphate that goes on through the rest of glycolysis. So let me back up and repeat because it's important that you are clear on this. Fructose 1,6-bisphosphate is cleaved, and that happens by the enzymatic action of aldolase, and we make two 3-carbon molecules. One of them, glyceraldehyde 3-phosphate, can continue on in glycolysis. The other molecule, dihydroxyacetone phosphate, can be converted to glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. So effectively, we have two glyceraldehyde 3-phosphates that continue on in the rest of the pathway. And so that's why I'm showing the 2x, is to indicate that everything I'm showing from this point on happens twice. The next enzyme in this pathway is glyceraldehyde 3-phosphate dehydrogenase. It catalyzes the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. As this occurs, we form NADH. So please keep that in mind. You've been seeing the participation of NAD plus and NADH several times, and this will really make sense when we finish our discussion of the metabolism of carbohydrates. So please note that we form an NADH at this point. Now the 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. So it's giving up one of its phosphoryl groups. The enzyme that catalyzes that reaction is phosphoglycerate kinase. And in this reaction, we have formed ATP. Because this happens twice, we actually form two ATPs. Now the product of this reaction, 3-phosphoglycerate, can undergo a mutase reaction catalyzed by the enzyme phosphoglycerate mutase, and we form 2-phosphoglycerate. 2-phosphoglycerate can undergo a dehydration reaction, which is catalyzed by the enzyme enolase, to form the product phosphoenol pyruvate. Phosphoenol pyruvate can then undergo a reaction with the enzyme pyruvate kinase and form the molecule pyruvate. In the process, we have formed another ATP, and because this happens twice, we have formed two ATPs. So let's take a step back and think about this pathway. We started off by spending an ATP in the first reaction, the hexokinase reaction. Then we had to spend another ATP during the third reaction, the phosphofructokinase reaction. So we had to prime the pump. We had to invest some cellular energy currency, ATP, to get the pathway going. But because everything happens twice from the formation of glyceraldehyde 3-phosphate on, we form 1, 2, 3, 4 ATPs. So we have formed a net of 2 ATPs. We can consider what we've been talking about with the aid of another figure, and that figure is shown here. Now you have access to this figure, and so we will let you work through this figure on your own. So now we're ready to tie together some loose ends and to summarize our discussion. We've seen that the net reaction of glycolysis is, we start with 1 glucose, 2 NAD+, plus 2 ADP, and we form 2 pyruvate, plus 2 NADH, plus 2 ATP, plus 2 protons. We also saw that the first step of glycolysis converts glucose into glucose 6-phosphate, a negatively charged molecule that is trapped in the cell. We learned that 2 ATPs are required to prime the pump and start glycolysis. These steps occur during the first reaction, the hexokinase reaction, and the third reaction, the phosphofructokinase reaction. Finally, we saw that 4 ATPs are formed, and thus a net of 2 ATPs, along with 2 NADHs and 2 pyruvates. We will consider the fates of these molecules shortly.